The overall goal of this experimental protocol is to provide a non-invasive and technically non-intensive procedure for inducing bacterial pneumonia in mice and the subsequent quantification of the in vivo host defense response in the lungs.
This method can help answer key questions in the fields of infectious disease and immunology about the genes and molecular pathways that control the host response to infection. The main advantage of this lung infection method is its technical simplicity, which allows even laboratories with little expertise in pulmonary procedures to study the pulmonary innate immune response.
In a biosafety level 2 facility, thaw a glycerol stock of K. pneumoniae and transfer 1 milliliter of the bacteria into a 500 milliliter culture flask containing 50 milliliters of tryptic soy broth. Incubate the suspension culture overnight at 37 degrees Celsius and 200 to 225 RPM. The next morning, dilute 1 to 5 milliliters of the bacterial culture in a new flask containing 50 milliliters of tryptic soy broth and incubate the culture in the shaker at 37 degrees Celsius for an additional 2.5 hours to achieve log phase. At the end of the second incubation, transfer 1 milliliter of bacteria from the second culture to a 1.5 milliliter tube for centrifugation. Remove the supernatant without disturbing the pellet, and gently resuspend the bacteria in 1 milliliter of sterile saline for 3 separate washes. After the third wash, resuspend the bacteria in another milliliter of sterile saline and set up logwise serial dilutions of